the airy silkworms are not. For that we have to do study. And since it is RNA virus, it's a little tricky. Always extract the RNA, we have to develop cDNA, then we have to do RT-PCR. Whatever bands we have seen, no, all are RT-PCR based bands. Yeah, as you could see here. So, these are the airy silkworm larvae. We fed this larva with virus. This is a pupa, moth and egg. We have extracted RNA because if virus is there, obviously you will get this virus RNA also. Then cDNA we have synthesized, and these are the bands. Interestingly, there is no virus band here. That means this virus does not have any cross transmissibility, which can able to infect only Muga silkworm. Go ahead. Same thing we have checked the cross infectivity in Bombyx mori, because in our nation, in our country, 90% silk production is from this Bombyx mori, right? India and China is the leading producer, and Karnataka, Tamil Nadu and Andhra Pradesh is the leading producer of this particular silk. And we are now interested whether Bombyx mori is susceptible to this hypovirus or not. The same technique we have applied, we have fed it, then we have generated cDNAs and we have seen. Interestingly, here also there won't be any hypovirus, except only in the control virus only we can able to see. There is no banding pattern in Bombyx mori, luckily. That means that this virus is highly specific to host strain. But in baculovirus, we have seen rod-shaped virus particles. They are not like this. They have the cross transmissibility. But this is very peculiar virus which is specific to only the wild silkworms. So when you are talking about wild silkworm, these all are lepidopterans. Go ahead. Then we are interested to see whether lepidopteran and other insects are susceptible or not. This is a Bihar airy caterpillar, one of the agriculture pests. We have found that this virus is unable to infect this. Go ahead. This is a semi-looper, one of the pests in castor. Okay. There is also, we could not find the expected band here. It is not at all affecting, although it is a lepidopteran insect. Then Spodoptera. Spodoptera is one of the famous agriculture pests. However, this is also free from that virus. So with this etiological studies, we could able to see this. This virus is highly specific to Vanya silkworms, which does not affect another lepidopteran. Yeah. Then, so we have identified the virus, developed certain diagnostic methodology. We have studied cross transmissibility. Then we could able to develop certain control measures. So for that, what happened? We have selected these chemicals and we have applied on this particular virus. Go ahead. The results indicated that slack lime, that is calcium hydroxide at 2% and bleaching powder at 2%. If you can mix it, it is more effective. That means that if you have a virus crop loss in the rearing field, we have to apply this particular reagent in order to save the crop, right? This is known as control measures. Okay. So we simply apply these chemicals on the polyhedral bodies, whether the polyhedral bodies are able to dissolve or not. Once you are able to get the dissolution of all polyhedral bodies, we can recommend the farmers, you go through it this type of protocols. Then only they will able to sustain in the field. Yeah. Similarly, am I going fast or what? I will ask you 2 or 3 questions, okay? You have to tell. So please pay attention. This is another insect that is known as Antheria mylata, tassar silkworm. In the tassar silkworm, these are the few of the symptoms, virosis symptoms. As in Muga, earlier we were not knowing this, which virus is causing this type of crop loss. Okay, please go ahead. While we were searching in the wild silk base, this is a database where you could get lot of sequencing information of wild silkworms. Fortunately, we found a signature sequence. This is actually a transcriptome, RNA level sequence of the tassar moth. This is actually not a host. This is a virus sequence. How come a virus sequence came into host sequence? That is our interest. So then immediately we have designed primers. Please go ahead. Then we have developed a PCR protocol assay. What is this virus? This virus is known as IFLA virus. This is single stranded RNA virus. The earlier one is hypovirus, that is double stranded RNA virus. This is a single stranded RNA virus. Then immediately we have actually deduced the genetic entire genome of the virus. The entire genome is 5' and 3' end. It's around 9,700 base pair. The entire virus genome is this. We have again 5' end, 3' end. At 5' end we have structural proteins. VP1, VP2, VP3 like this. At 3' end we have RDRP. So, established the genome organization of the antherium myelifla virus. Please go ahead. Then again, as you could see here, this is actually a phylogenetic tree. Again, it is showing antheria prioli ifla virus. That means that the ifla virus, which is affecting myelita and prioli, are both are same. It is further confirmed with the different type of tissues. The virus is present in all parts of tassar silkworms. As you could see, fat body, midgut, silk gland and all, these are different body parts of insects. All body parts we can find. That means that this is a systemic infection. There are two different kinds of viral infections. One is localized, the other one is systemic. Systemic means you can able to find the virus signature throughout the insect body. Localized means only in one particular tissue only we can be able to see. So this particular one, sorry. Then we have done in which body parts this virus will be more, like where it exactly multiplies more. So for that we need to have a quantitative real-time PCR. As you could see here, the fat body and silk gland, we have more multiplication of this virus. The same virus we have find in another silkworm, Antheria prioli. But interestingly, in Antheria prioli, the same virus is present along with another virus. That means there are two different kinds of virus we can find at a time. That is known as latent infection. So one is ifla virus IV, then is NPV. Yeah, thank you. This is my team. He is our HOD, head of the department, Dr. Ponnuvelu sir. This is my team. These two are bioinformaticians. We have very good infrastructure to do bioinformatic analysis. We have power stations and servers are there. So my collaborator at Northeastern region, this one, Indra Kumar, is a Tamilian guy, and these two are also working in the Northeast because our institute is located at Bengaluru. We don't have Moga silkworms here. So if you want to work with them, so we have to make collaboration with other scientists in the Northeastern region. And she is my student, she is doing PhD in this. The second part of work, especially ifla virus, this work is done by my in charge, Ponnuvelu sir, with collaboration with a Swedish counterpart. He is only 10 years from Swedish Agriculture University, Uppasala. So there are certain techniques we can't do by ably, collaboration is very much required. So we are into this only 10 years from Swedish University, we work together. I would like to express my sincere thanks to Neethrajan sir and also Shobhana Ponnuvelu madam for giving me this opportunity to share few of my research findings with students. Thank you. Yeah.
Thank you, sir. Thank you for that wonderful presentation and thank you for enlightening us with that talk.
Any questions, if they want any questions?
The session is now open for discussion. Students, if you're having any questions, you can ask now.
Hello, sir. Sorry. Baculovirus is here, sir. Yeah. Baculovirus is acts as a microbial insecticide. So it is used in the sericulture practices like baculovirus. It is acts as a microbial insecticide.
Yes, sir.
It is used in sericulture practices.
No, sir. Baculovirus are very much dangerous to silkworms. So we have actually baculovirus diseases in the sericulture. It's a major problem. That's why we are identifying which baculovirus is that. So we are developing control measures. So that is the big enemy for sericulture. So
Another question, sir. Double stranded DNA or single stranded RNA, which one is the highly virulent virus, sir? Single stranded DNA or single stranded RNA, which is the highly virulent virus?
DNA, sir. Single stranded DNA is the most virulent. Single stranded RNA will give you a chronic infection. Immediately silkworms may not die. Okay, it takes so much time actually. So, but single stranded DNA is lethal. We can find within 4 days or 5 days we can see the crop mortality, especially Bidenso virus. Bidenso virus is having single stranded DNA, so it gives you 90% mortality.
Thanks.
I have one more doubt, sir. You have not taken it to the protein level. As
Yeah.
I can see, there is no structure for all these viruses. So you have any future idea of taking it?
What, madam?
So you have done all DNA tests and other tests. Only one protein test was only done. So you have just seen whether the protein is there or not. So as of I can see, all this virus, some of the viruses has no structure at all. So what do you think of that perspective?
No, madam. Cypovirus, actually, we have identified this pathogen is a cypovirus, madam. Cypovirus has well-defined structures are there. It is actually spina rheoviridae. It has a spike proteins, which is caused by segment 1 and segment 2, actually. So here, in order to develop a diagnostic methodology, only polyhedron we have cloned. So then what happened? We have sent for our genome for sequencing. Once you get entire genome, then immediately we will come to know the proteins and all, madam.
Thank you, sir. Thank you.
Good afternoon, sir. I have one doubt that only virus will affect the silkworms or a virus affecting bacteriophages there, no, sir? Like the bacteria will also cause any viral disease in silkworms, sir?
No, virus, each virus has a specific host. So this virus is affecting silkworms, right? So bacteriophages, matlab, they are the viruses which affects only the bacteria, not the silkworms. So bacteriophages, the host agent is different. That is bacteria, okay? They cannot infect silkworms.
Only virus will affect or any other parasites?
Yes. Yeah, I will tell you. Generally, we have 4 different kinds of diseases we can find in sericulture. So one is virus. Okay, we have seen all these things. Then bacteria. We can Bacillus thuringiensis, Streptococcus species, some Bacillus ameloleukophaciens, and Serratia marescens. Okay, and Klebsiella, Enterococcus. These are different kinds of pathogenic bacteria. This is bacterial disease. The first one is virus disease. The third one is fungi, muscardine. So you may be knowing, entomologist, muscardine is caused by different kind of Beauveria bassiana or Metarhizium species. And the fourth one is Nosema species, that is caused by, the disease is pebrine. Nosema is pathogen, bacteria is pathogen, fungus, muscardine is a pathogen, and virus is the pathogen.
Yeah, sir. Thank you, sir.
Good afternoon, sir. Sir, like BT cotton, can we able to produce any variants in mulberry leaves like that can kill the viruses?
To kill the virus, see, generally, this is RNA, right? In order to break down this RNA, we have to silence that particular RNA molecules. So we are doing what? Basically, there is siRNA, miRNA. These are the small RNA molecules. Will go and bind your targeted virus and they will degrade. So we can use transkingdomal approach. So we can express those miRNA, a small iRNA into this silkworm. Then only we will be able to save the virus. Sometimes what happen? See, I'll tell you. There are 2 silkworms. One silkworm is susceptible to the virus, but the other silkworm is not susceptible. That means the genetic content matters, right? So first we segregate which are resistant to this virus, right? So for example, we have collected 4 silkworms. All 4 are resistant to the virus. Then we make a breeding approach. So we want to develop virus resistant breed. So that type of work is also we are doing that.
Thank you, sir.
Sir, this is a very general question, and I know we earn a lot of money from it's a very commercial product, but why are we torturing those small insects? Don't we have other means of producing silks other than only the worms, relying on the worms and killing them? Is that the only way, or are there other means? Because we have developed technically very far, no?
Madam, this is very nice question. Actually, what happened? Nowadays, Central Silk Board is also concerned a lot about this. Now we are coming up with a technique known as Ahimsa silk. So Mahatma Gandhi is our role model. So what is meant by Ahimsa? Basically, the cocoon we have seen, no? The second silkworm, eri. So no need to cook those cocoons. We cut the cocoons, we remove the pupae, whatever the cut cocoons, we spun the silk and we do that. So no, the pupae will be useful for seed generation. We will use this pupae, we will keep it like this. After 10 or 15 days, we will get a silk moth. So they couple each other and produce eggs. These eggs we will sell. So the complete life cycle we will follow. Especially, madam, Jain people and Buddhism, they won't wear silk. They think that silk is coming because of killing these silkworms like this. So that's why now we have started that, madam.
Students, if we are not having any questions, we shall move on to the last session of the day. This is valedictory function. Yes? Yes. Thank you so much. So now we are moving on to the last part of the program, the valedictory function, and it will be chaired by Mr. Yuvraj sir, Head Department of Microbiology. I am handing over the session to Dr. Yuvraj sir.
Thank you, ma'am. Good evening, ladies and gentlemen. We hope that you had a wonderful time in this DBT-sponsored first international conference on the significance of gain and loss of function and impairing transcription factors causing genetic disorders in humans, and enjoyed our hospitality too. Setting goals is the first step in turning the invisible to visible, says Tony Robinson. Our goal is to attain a prosperous valedictory function. We humbly believe that we achieved our objective and as well as the goal of our conference. Yes, ladies and gentlemen, we are at the brink of this international conference. This gathering is reconvened to witness the auspicious valedictory function. Pleasantries. Honoring the guests always the traditional practice of our culture system. We are happy and humble to recognize and honor the guest on behalf of our management, for which I am delighted to offer a happy welcome to and as well as the invitation to our Professor, Principal Sir A. Muttumani, to honor our chief guest of the valedictory function, Dr. Gangavarappu Subramaniam Sir. Thank you, sir. This culture system is known for its reception. We Indians greet all with our smiling faces, knowing that all the humans are nothing but the embodiments of divinity. To welcome this august gathering, I kindly request Dr. G. Kirtana, member of the organizing committee of this conference, to propose the welcome address.
Thank you, sir. Yeah. A warm and a cheerful noon to everyone present here. We are pleased to welcome everyone for the valedictory function of DBT-sponsored International Conference on the Significance of Gain or Loss of Function and Impairing Transcription Factors Causing Genetic Disorders in Human. With immense pleasure, we welcome our dearest Principal, Sir, Professor Dr. Muttumani, Sir. Welcome, Sir. Distinguished Chief Guest, Dr. G. Subramaniam, Sir. Welcome, Sir. We render a warmest welcome to respected head and faculties of various departments, distinguished researchers, world, dear colleagues, and dear my students. I welcome you all on behalf of PG and Research Center in Biotechnology. Welcome everyone. Yeah. And my heartiest welcome to our eminent speakers, Dr. P. Sundaresan sir, Dr. G. Muthusamy sir, Professor J. Martin Collinson sir, Dr. Akshaya Murugesan, Dr. Arjun Masurkar, Dr. Ruby Vijay Kumar, Dr. Satish Kumar, and Dr. G. Subramaniam sir, who had shared their invaluable time and knowledge with us. I welcome all the guests from different colleges and universities from different parts of the world. We hope that the two-day conference would have helped the participants to expand their mental horizon and enrich them with knowledge from all around the world. We welcome our media friends who are very supportive these 2 days. Welcome media friends, and once again, we welcome you all for the valedictory function. Thank you.
Thank you, ma'am, for a nano-sized welcome address. I kindly invite Dr. P.S. Shobna for a pleasant task.
Hello everyone. I'm here to introduce our chief guest, Dr. G. Subramaniam. He's a scientist C from Central Silk Board, Bangalore, Karnataka. He has done his post-graduation in microbiology in College of Science and Technology, Andhra Pradesh. He has done his PhD in Department of Microbiology and Biotechnology in the Maharaja Sarojirao University of Baroda. He has served as a principal investigator in 4 projects and co-investigator in 6 projects. He has also won several international and national awards. He has presented several international and national seminars, workshops, training courses. And here, thank you for coming, sir. I thank Principal sir and HOD sir for this opportunity. Thank you, sir.
Thank you, ma'am. The interaction of knowledge and skills with experience is the key to learning, says John Dewey. From the bottom of my heart, I render my cheerful welcome to this experienced stalwart, man with voluminous experience, chief guest of this valedictory function, Dr. Gangavarapu Subramaniam sir, Scientist C, Seri Biotech Research Laboratory, Central Silk Board, Government of India, for his valedictory address. Sir.
Honorable Principal Sir, Muthumani Sir, MGR College. Okay. And respected Dr. G. Nithirajan Sir, Head of the Department Biotechnology, and also Convener and Organizing Secretary for this 2 days international program. Dignitaries on the dais and off the dais, dear student friends and participants, a very good afternoon. Yes, a very good afternoon, one and all. Yeah, we had already interactions in my deliberation. However, I have few points. The first one, we know this cell. What is cell? Cell is a structural and fundamental unit of a life. In mammalian species, we have approximately 200 different kinds of cells. These all cells are highly heterogeneous in nature. Their structure, their shape, as well as their functions are very unique. What is the driving force which actually conferring this particular type of special phenomena? So that is nothing but this transcription, initiation factors. Transcription initiation factors actually decides which type of proteins has to express or it has to be repressed. They do a lot of moral policing in our body. So obviously, in the first day, Sundaresan sir was telling, he was pointing out central dogma of life, DNA to RNA, RNA to protein. So RNA to protein is one of the vital step in our body, in our cell. So that is nothing but a protein synthesis. But this protein synthesis is highly regulated phenomenon. It's tightly regulated by transcription factors. Since I must tell you, everyone knows what is the size of human genome. If you can anyone tell, what is the size of human genome? Anyone? Please raise your hand. Should know, students know. Biotechnology. Yeah. Sorry. It's around 3 billion base pairs. Though it's a vast genome, we have 20,000 different kinds of protein in our body. 20,000 proteins. Among these 20,000 proteins, 2,000 proteins are coded by particular genes that is known as initiation factors, transcription initiation factors. So total proteins are 20,000, 2,000 are your transcription factors. So that means 10% of our human genome is actually coding transcription initiation factors. So you should realize the importance of this transcription initiation factor. On the other day, I'm new to this subject basically, I'm a virologist. And the other day I was actually reading few of the articles. What happens if any find mutations or any defects in this particular transcription factors. So it's very surprising to see this. Almost all the chronic inflammation, whatever we face in our post-COVID scenario, all is due to this post-transcriptional factors. So asthma, it's responsible by transcription factors. Different kinds of cardio, diseases, again same. Defects are nonsense or missense mutations in transcription factors. And again, we have seen a lot of NVIDIA. So I must congratulate Nithirajan sir. He has done a substantial research, especially this PAX6 and FOXA, something like this. He has done a lot of work how this transcription factors is actually involving in NVIDIA, congenital NVIDIA, means eye development and eye disorders, retina manifestations and all. So your eye is deciding by transcription factors. Brain is actually controlled by transcription factors. Heart, liver, in whatever disease you name it, largely or to some extent is actually influenced by the functioning of transcription factors. If the genes are very good, there is no mutations, everything will be fine. There could be a good equilibrium. Homeostasis will be there. But if anything is defected in the transcription initiation factors, obviously we will end up with chronic or maybe acute diseases. So I think that's the reason he has selected this topic. And this is a highly advanced and very specialized topic. For selecting such a wonderful, very much required in the hour. So I must congratulate the college as well as the principal sir. However, nevertheless, to do such type of studies, we need to have, first of all, finance, funding, and also good infrastructure. Most importantly, we need to have collaboration out of borders. Okay, then we can do very good research. And I must say that we at Central Silk Board, especially in Cerebiotech Research Lab, you please visit us. We have a very good infrastructure, starting from genomics, proteomics, we started breedings, I am doing virology and all. Please visit us. If you need any help, we will be very happy to support you. We are always with you. And also, I thank the principal sir, Nithirajan sir and Sobna madam for giving me the opportunity. And I request students, the science is such a vibrant field, should have very constructive and positive mind and kindly continue your career in the scientific field. You are the future of our nation. That's my request. Thank you one and again. Jai Bharat.
Thank you very much, sir, for the invitation, and surely we will disturb you in this regard always to gain academically whatever the information we need. Thank you very much, sir. The organizing committee of this conference believes act of giving something to others is an art of flowering your heart. Presentation or oral presentation, best 3 oral presentation are identified and recognized with a shield and certificates. To distribute the certificates to the winners and the participants, I feel honored in offering a cheerful welcome to the dignitaries on the days to come forward to distribute the certificates. Best 3 oral presentation. The third prize goes to Mr. Tarun Kumar and Kiran Kumar, BTech Biotechnology, Adiyaman College of Engineering. Second prize goes to Ms. C. Divya from Periyar University, Salem. Second prize, C. Divya from Periyar University, Salem. And the first prize goes to Dr. Lokesh ST from Kuvembu University, Bengaluru. For getting the certificates from college-wise, I read out a representation from each department. Kindly come and collect. Ms. Mary Blessy, Department of Biotechnology, Adiyaman College of Engineering. Followed by Mr. Tirunavukarasu, Department of Chemical Engineering, Adiyaman College of Engineering. Mr. Arul Kumar K from Periyar University, Salem. Arul Kumar K. It's not there. Okay. Arun R from 3rd BSc Microbiology, MGR College. Arjun C, 2nd BSc Microbiology, MGR College. Archana S from 3rd BSc Biochemistry. Biochemistry of MGR College. Any representation from Biochemistry? No. Okay. Yes. Come fast, please. Settle down. Kiritika GV, first BSc Chemistry, MGR. Thank you, sir. I read out the colleges who are going to get the certificate in absentia. St. Joseph College of Arts and Science for Women, Hosur. Aringaranna College of Arts and Science, Krishnagiri. Sona College of Arts and Science, Salem. Batmavani Arts and Science College for Women, Salem. Mutayamal College of Arts and Science, Rasipuram. Christ University, Bengaluru. The Oxford College of Science, Bengaluru. Jain University, Bengaluru. Christ Jain College, Bengaluru. Reva University, Bengaluru. AMC College of Bengaluru. An appeal to all the winners. Happiness is like a perfume. You can't spread on others without getting a few drops on it. So always be happy to make others happy and make your knowledge infectious for the betterment of the society. Opportunities are equal for all, but feedback? No. Gratitude is not only the greatest virtues, but the parent of all. To record our gratefulness, I would like to invite Dr. R. Muthuswamy, organizing committee member of this conference, to propose the vote of thanks.
Very good evening to all. I deem it a great honor and a privilege to propose the vote of thanks on these memorable occasions. So let me first of all start by giving glory to the Amity Good for making today occasion a resounding success. So I would like to thank our chief guest, Dr. Akshaya Murugesan, and then Arun V. Murugesan sir, and then Ruby Vijayakumar ma'am, Satish Kumar Rajashekaran sir, and Sundaresan sir, and Govardhan Muthuswamy sir, and Dr. J. Murugan sir, and Govardhan Muthuswamy. And also, I would like to thank Dr. Gangarajupu Subramaniam sir for his wonderful presentations. My heartfelt thanks to all the head of the departments, various departments, and faculty members, students from various departments, and also my special thanks to our own departments, biotech department students. So without their contributions, we cannot finish this conference, other supports during the preparations and arrangements all. Especially my sincere thanks to third years and even second years, all the students. And also I grateful to thank our management and other non-teaching staff members. And finally, I would like to thank our principal, honorable principal, and then our organizing committee members, Nidhi Rajan sirs, and the media peoples. So without their support, so we cannot export these conference throughout the world or everywhere. So I finally thank our media peoples, and also some of the support members, for example, for giving the current supply, everything. That means for non-teaching staffs. So thank you for giving this opportunity. Thank you one and all. Thank you.
Thank you, sir. Before concluding, I kindly request you all rise for the national anthem.
Janagana Manadinayaka Jaya He Bharata Bhagya Vidata Punjab Sindh Gujarat Maratha Dravida Utkala Banga Vindhya Himachala Yamuna Ganga Uchchala Jalati Taranga Tava Shubha Name Jahe Tava Shubha Shisha Mahe Gahe Tava Jaya Gada Janagana Mangala Dayaka Jaya He Bharata Bhagya Vidata Jaya He Jaya He Jaya He Jaya 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 He.
Life is about the people you meet and the things you create with them. Hope we will meet you once again in second international conference with us next year. Until then, ma salama from the team. It means goodbye until we meet again in Arabic. Thank you very much. Thank you. And the most important time, as you know that, the entire international conference is sponsored by DBT, Department of Biotechnology, Government of India. On behalf of the convener and as well as the management and our principal sir, we wholeheartedly thank DBT for the wonderful support. Thank you. Okay. The other college students can leave, and as well as the department too. Department of Biotechnology students of MGR College are asked to stay in the hall. Remaining, you can leave. Thank you.
PG Biotechnology students, please come on the stage. PG Biotechnology student, please come on the stage. PG Biotechnology for first MSc and second MSc students, please come on the stage. Come fast. Come fast. Finally. Final. Yes. Come fast. PG students, come fast. உள்ள வாங்க, ஏன் போட்டு நின்றுது? உள்ள வாங்க, இந்த பக்கம் பாஸ் முன்னாடி. யாரும் இல்லப்பா. Get ready for third year students. Third year students, get ready. Boys, third year students, please come on the stage. Third year students. Third BSc boys. Third BSc boy students, please come on the stage fast. Third year girl students, ready? Cover ஆகுதா? Girl students. இப்படி போங்க இப்படி போங்க இப்படி போங்க இப்படி போங்க. டேய் தம்பி. Fast. Come fast. Second year. Second year students, get ready. Second year students, both girls and boys. Second year students, come fast. Second year students, come fast. First year boys, get ready. First year boys students, get ready. First year boys students, get ready. கொஞ்சம் உள்ள தள்ளிக்கப்பா. ஏன் இப்படி போங்க? பயப்படுறீங்களா? உள்ள தள்ளி வாங்க.